Thank you. So, hello everybody. My name is Kalifa Samake. I'm working now on TLRs. I would like to talk a bit about those work we are doing on TLRs. And we actually decide to present again this work here because we think that it's relative to the topic of the conference. So let me move to the presentation. I will start by introducing TLR and saying that TLR are key component of the immune system, and they play a key role in the immune system. That's why they are considered as prospective target for health breeding in livestock species. TLR are in the beginning of the toll-like signaling pathway. In cattle, they have divided in two paralogs. Okay, so the protein product consists of inner region, which is the extracellular part of the protein, and the transmembrane part, and also an intracellular part. So we all know that the TLR diversity in livestock species has been characterized in many projects and associated paper. However, it is necessary to determine the haplotype structure of the known SNP in order to solve the effect observed in association studies. Therefore, we explore the haplotype structure of antibacterial TLRs. So TLR gene coding for toll-like receptors of antibacterial series, namely TLR1, TLR2, TLR4, 5, and TLR6, were resequenced using the technology of Pacific Biosciences, and also the whole genome sequencing with Illumina X10 technology was applied in order to reduce the error rate of sequencing and to confirm the SNP for subsequent genotyping individual animals. Yeah. So the SNP present in the population were established within the range of the designed Pacific Biosciences amplicon, especially in TLR2. In TLR2 and, sorry, yeah. In TLR2, the network graph for the reconstruction, for the reconstructed haplotype structure, which you can see here, was present in FANG during FANG conference at the beginning of this year. And surprisingly, we find there 15 haplotype in amplicon 1 located in the proximal part of the TLR2 transcript. Those 15 haplotype were forming two, or are forming two distinguished groups in order in other ways, in other words, they demonstrate a binodal distribution of frequency. When we use all 11 SNP for designing the network graph, we observe such graph. But when we use only seven SNP, the graph were also a bit different, but here the binodal distribution was clearly observed. So on the other hand, when we also look at other TLRs with a similar binodal distribution was detectable in TLR5. Here you can see it on your left, and TLR6 here on the right, at the proximal part of, at the proximal region of TLR5 transcripts mainly. So on other hand, also an unclear trend of binodal distribution was observed in TLR4 and in TLR5. Sorry, in TLR4. And okay. Surprisingly, this similar results were reported in previous clustering of TLR, which were published in by a group of researchers from Turkish in the historical bridge. However, the origin of this disequilibrium is still not explained. So actually, we are trying to give some hypotheses which can be the possible explanation of those of this disequilibrium in the haplotype structure. The fluctuating selection by alternating infectious agent have been suggested by Bilgen and to explain this disequilibrium. However, one of the haplotype group should be sooner or later lost either by the prevailing infection pressure or as a result of the stochastic process. Sorry. We suggest that the balancing selection might be originate from two different essential functions performed by the TLR genes or their product. An example of the dual function might be the formation of two kinds of product differencing in specificity, like the interaction of TLR2 product with TLR1 and TLR6 that form functional dimer that in their turn recognize different ligand of bacterial origin. In case of the heterozygotes advantage mechanism, the recombination should lead to the merging both haplotype groups. The formation of two stable groups of haplotype in TLR2 can also be a consequence of disruptive selection. On the other hand, the association of the observed group of haplotype in the proximal region of TLR2 and TLR5 transcript suggests that the selection target might be located in the non-coding 5' region of those genes. In conclusion, I would say that we are planning to design other amplicon which can be used on the platform which have low error rate of NGS, and we are also planning to for more reliable and efficient genotyping using snapshot or similar methods. So that's all from my side. Thank you for your attention. And if you have any question, I will be happy to